Hello everybody and welcome back to the Sanctuary. I'm your host Professor C, and we're going to do some more A&P today. Specifically, we're going to do a little short lecture on smooth muscle and how it differs from, say, skeletal muscle, which we primarily focused on. So let's do it. Alright, the first thing we need to do is to remind ourselves that the anatomy of a smooth muscle fiber is way different than a skeletal muscle fiber. If I could draw a skeletal muscle fiber up here, I could draw part of it. It kind of looks like a very long pipe, and it would extend off the screen, and it would have this banding pattern that's very evident, and we'd call those striations, right? That's what we spend most of our time talking about in the last few talks, is skeletal muscle. But remember, smooth muscle is much different. If I can outline one here on the left, it looks very much different than a big long pipe. It looks kind of like a UFO from, say, a 1950s comic book or something like that. So first of all, they're extremely short compared to the very elongated skeletal muscle. Some cite that it's 1,000 times shorter than a skeletal muscle fiber. They have tapered edges, that is, they're kind of a bulging middle, which it should say in a moment, but these edges taper off to a point on either side. And there's one single nucleus. Remember that skeletal muscle, cardiac muscle, was multinucleate, so we should see a single bulging middle with the nucleus inside. Again, no striations here, and we're not going to see any myofibrils. If you remember, again, what a skeletal muscle fiber looked like, you could just look up here at the top left of the screen. Remember, we had all these little tiny, looked like a pack of circular cigarettes, right? Those are all myofibrils. We don't have those here in smooth muscle. All right, if you were to look inside of the cytoplasm of a smooth muscle fiber, and I'll kind of do it like this, you will find actin and myosin. So that is not anything different, but the way it's arranged is quite different. You'll see it kind of anchored to one side of the membrane, and you'd see some actin and some myosin and some actin and some myosin and some actin and some myosin, and then reach all the way over and anchor to the other side. And then maybe you'd see another example here with some actin, with some myosin, and some actin and some myosin overlapping in a similar fashion all the way across there. And you see these strings of overlapping actin and myosin. There's no Z-lines as we were thinking of back when we saw sarcomeres. So no Z-disc. They will sometimes have structures called dense bodies to which they're attached to the membrane, but we're kind of skipping over that. There is no such thing as troponin. If you can remember, troponin was a regulatory protein, and if calcium were to bind to troponin, it would cause the actin to twist and allow myosin to bind to it. So it doesn't have that same regulatory protein. It could have something like calmodulin or some other ones, and there's a whole slew of these that are substitutes for troponin. Smooth muscle contracts involuntarily. You're not in control of how your smooth muscle contracts. And it does so because of this here, I'm drawing these arrows, because of those overlapping strings of actin and myosin. When they contract, they will contract in a strange kind of a corkscrew fashion, more than we saw just a linear fashion from the skeletal muscle. Okay, there are two types of smooth muscles that are usually talked about in an A&P course. That would be single unit and multi-unit. So this is introducing single unit smooth muscle, and it is the one that most people think about when they think about or hear about smooth muscle for the first time. It's also called visceral muscle. It would be lining the walls of tubular or hollow organs, and we see a lot of examples right there. We see some of the small intestines, we see some of the large intestine up here where I'm scribbling my pen, and we see the stomach here, which is also a hollow organ. Now, an exception is, of course, the heart. The heart is cardiac muscle, and we'll get to the heart much, much later in another chapter. So these single unit smooth muscle systems have many, and I'm going to put this many here just so it stands out. They have many gap junctions, and if we remember from way back in some previous lectures, gap junctions are kind of like electrical connections between two cells that lets them share information, right, to pass on action potentials potentially very rapidly. So we're going to have a system here where there are many of these single unit smooth muscle cells, and they're going to form a syncytium, is how to pronounce that word. A syncytium is a collection of things that work as a unit. So we will get this system where we have a lot of electrical connections, and when one fires, it kind of tells everybody else around them to fire, almost like the way that a flock of birds coordinates through the sky. So they have many gap junctions that form these functional syncytia that work in some sort of coordinated manner to contract or relax the tissue. This is single unit smooth muscle. Now, single unit smooth muscle is referred to as myogenic muscle, meaning that a lot of the tissue itself creates action potential spontaneously. We don't have to actually give it signals from the nervous system. Sounds strange to hear at first, but the heart can do that as well, right? It's got its own timing mechanisms. So the term myogenic means, again, muscle for myo, and genic from genesis, beginning or originating from. So myogenic means that the spontaneous action potentials are generated from the muscle tissue itself, not the nervous system. Now, this squeezing, this myogenic action of this smooth muscle, pretend you have something here, and we'll just pretend it's a ball of food or something. And what you would do is, to move that through the tubes, you would have this part of the tube, the intestines, squeeze. That would be the duodenum there, right? You'd have this part of the intestine kind of open up, would relax. And then you'd do the same thing. You'd kind of squeeze it and then relax it and then squeeze it, and slowly, 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 slow and steadily, right? This will make its way down the length of the pipe. And you want to do this in the intestines because as the food moves through here, you want to rip it apart, digest it properly, absorb it into the wall so you can use it later. So you want a slow and steady race, not something like a skeletal muscle, which would push it through extremely fast. So this is called peristalsis, this slow, steady movement, the slow contract, slow relaxation that slowly squeeze substances through the lumens of these hollow organs, kind of like toothpaste moving out of the tube very slowly. Okay, multi-unit smooth muscle, and what bothers me here is it doesn't have the hyphen in between, and it looks a little confusing to me at first. But multi-unit smooth muscle is much different. We find it in limited areas. We don't find it like lining the hollow organs like we saw with the single units. So we find it like in the throat and the trachea, the windpipe, if you know that term, the iris of the eye, which we see pictured here on the left-hand side of the screen, the colored portion of the eye, and lining some of the walls of the large elastic arteries that are near the heart. These are strange because they act a lot like skeletal muscle in how they contract very quickly and then relax very quickly, but they're not served by the part of the nervous system we would think. They're served by autonomic fibers. And these are ones that, of course, are out of your control. They fire automatically, right? Even though the term is autonomic, they're an automatic system. So these are referred to as neurogenic. These multi-unit smooth muscle tissues are known as neurogenic. They have to receive information from the nervous system, specifically the autonomic nervous system, in order to do their squeeze and release, right? To show a picture of kind of what that would look like in a multi-unit system, we have these structures called varicosities, and basically they're just a series, one after the other, of autonomic neurons that have many branches and many of these bulbs or these expansions. And they're going to loosely form what we would call a motor unit if we were talking about skeletal muscle. But these are varicosities, and this is how autonomic signals get to that multi-unit smooth muscle system. All right, a pretty quick one. Thanks for watching that one, of course. Check out some more videos in this series if you want to see some more about muscles or anything else to do with A&P. See you for the next one. Bye-bye.